and the parathyroid gland followed by the histology. So thyroid gland is a shield-like endocrine gland. Okay, it is a shield-like endocrine gland having rich blood supply. It is situated in the lower part and in front of the neck and the side of the neck. Lower part of the neck in front and side of the neck. Now, the thyroid gland is having two lateral lobes. Okay, two lateral lobes are there connected by isthmus. Okay, that is the structure of the thyroid gland. Sometime an additional lobe is extending upwards from the isthmus. It is called pyramidal lobe. Okay, and from the isthmus, a fibromuscular band is extending up to the hyoid bone. It is called levator glanduli thyroidea. Now the thyroid gland, and on the posterior aspect, or you can say on the posterior border of the lobes of the thyroid gland, the parathyroid glands are situated. Total 4 parathyroid glands are there. Okay, 2 pairs of parathyroid glands. Next, if you see the thyroid gland, the main function is to regulate the basal metabolic rate, stimulate the somatic and psychic growth of an individual, and plays an important role in calcium metabolism of our body. Okay, these are the functions of thyroid gland. Now, in calcium metabolism, parathyroid gland is also helping. How it is helping, we'll see. Next, there are some peculiarities of the thyroid gland that you have to know. First of all, the thyroid gland is the largest endocrine gland of our body. Okay, जितना भी endocrine gland होता है, among them thyroid is the largest endocrine gland of our body. Next, the thyroid, where exactly it depends. Now, when the production of hormone is taking place from thyroid, it depends on external source, that is iodine. Without iodine, it will not be able to produce any hormone. Okay, that means for production of hormones, it is depending on a external source. That is a unique feature of the thyroid gland, and that source is iodine. Okay, iodine is not produced in our body. We have to intake the iodine. Okay, from there the thyroid is producing the hormone. Okay. Next, this is the only endocrine gland which is placed superficial in our body. Other glands are situated deep, endocrine glands are situated deep in the body. So these are some important peculiarities of the thyroid gland. Next, here you can see in the diagram the parts of the thyroid gland. So first of all, thyroid gland is having a connective tissue capsule. Actually, true capsule, false capsule is also there. That we will study later on in the gross part. But though it is related to the histo, that's why I am mentioning that true capsule and false capsule both are present. Okay, as you can see in the diagram. Okay, I hope you can see the true capsule. Next, in case of thyroid gland, these are the lobes, these are the lateral lobes. Okay, you can see these are the lateral lobes of the thyroid gland, and this is the isthmus of the thyroid gland. From the isthmus, you can see an additional lobe. Okay, it is occasionally present. It is called as pyramidal lobe. Every time it is not present. In each and every case, it is not present. Next, you can see a fibromuscular band is extending. That is called as levator glanduli thyroidea. Okay, so this is the overall structure of the thyroid gland. Okay. Next, in the gross, you have to study about the surfaces, borders of the thyroid gland, relations of the thyroid gland, coverings of the thyroid gland, nerve supply, blood supply, lymphatic drainage, as usual. Okay. But now directly we'll go to the microscopic structure. Now in microscopic structure, suppose you have taken a section from here, or you have taken a section from here, then what you can see under microscope. Okay, so in the histology of the thyroid gland, first of all you have to understand one thing, that thyroid gland is covered by capsule, as I have mentioned. So if any structure is covered by capsule, then what will happen? The capsule will send septa, okay, inside the gland, and it will divide it into lobules. Okay, if you remember the previous structures which we have studied, so same thing is taking place over here. So here in case of thyroid gland also, dense irregular connective tissue capsule is there, okay, which is derived from deep cervical fascia. Next, you can see the parathyroid glands are also situated. So parathyroid glands are situated on the posterior aspect, okay, of the thyroid gland. When we will see the microscopic structure of thyroid gland, as I said, at that time, first of all, you will see that connective tissue capsule septa are there. In between the septa, lobules are present. If you take one particular lobule, then inside the lobule, what you can see? You can see that it is made up of aggregation of follicles. Inside the lobule, what you can see? Aggregation of follicles. Okay. Next, these follicles are nothing but it is forming or it is making the parenchyma of the gland. These follicles. Okay. Now, these follicles, the size of the follicles is 0.2 to 1 millimeter, and inside the follicles you are getting two things. First are the follicular cells, okay, and inside the follicle you will be getting a homogeneous pink color structure. Those are called as colloid. Okay, center of each follicle, gel-like acidophilic homogeneous material is present, that is called as colloid. Okay, so these are the two things which are present inside the follicle. Now, inside the follicle, the cells which are present, okay, it varies from simple squamous epithelium to simple columnar epithelium. That we will see why different types of lining epithelium are present. Okay, so first capsule, septa. Through septa, blood vessels, nerves are passing, going deep into the gland. Lobules are there. If you take one lobule, inside the lobule, what you can see? Follicles, multiple follicles. Okay. Now, inside the follicle, two things are there. First, you can see the follicular cells and gel-like acidophilic rich colloid. Okay, gel-like acidophilic pink homogeneous material, that is called as colloid. That is also present inside the follicle. Okay. Now, as I said, the follicular cells, it varies from simple squamous epithelium to simple columnar epithelium. Next, on H&E staining, the follicular cell shows slightly basophilic cytoplasm. Now, this you have to understand, because based on the staining difference or based on the staining properties, you have to identify the follicular cells. तो जो follicular cells है उसका appearance कैसे होगा? Okay, what will be the appearance of the follicular cells? So this shows slightly basal basophilic cytoplasm with spherical nucleus, okay, and one or two nucleoli. Okay, so basal part is basophilic in nature. That means it will be darkly stained. Next, interfollicular space is there. What is the meaning of interfollicular space? The space between the follicles. Okay, so in that numerous blood vessels, lymphatics, connective tissue fibers, all those things are present. Next, in the interfollicular space, we are getting the parafollicular cells, are also called as C cells. Okay, parafollicular cells are also called as C cells. Now, these parafollicular cells, either they are embedded within a follicle or in between the follicles. Okay. Now, the question is how you will identify the parafollicular cells. First of all, these are polyhedral cells, oval eccentric nucleus. Okay. Now, these cells, either they are single or lie in clusters. So, mostly these parafollicular cells are in the form of clusters. Okay. Now, parafollicular cells also shows numerous secretory vesicles. Okay. Now, what is the function? Follicular cells secrete two hormones, that is T3 and T4, also called as thyroxine and triiodothyronine, that is T3 and T4. And parafollicular cells secrete a hormone that is called as calcitonin. Okay. So, these are the functions of the cell. Next, you have to understand one thing, जो follicles होता है. Okay, as I said, the follicles, cells which are present in the follicles, every time it is not same. Amount of colloid which is present inside the follicle, that is also not same, it is different. Now, that depends on the activity of the follicle. Now, when the follicle is highly active, at that time it is lined by columnar epithelium. Okay, when it is highly active, it is lined by columnar epithelium. At that time, the amount of colloid is scanty. Moderate active follicles, it is lined by cuboidal cells, okay, with moderate amount of colloid. And if the follicle is inactive, at that time it is lined by simple squamous epithelium with abundant colloid. Okay, that means जो follicle होता है, follicle is having different forms. Some follicles are very active, some are moderately active, and some are inactive. If it is inactive, then what is the lining epithelium? Simple squamous epithelium. At that time, inside the follicle, huge amount of colloid are present. ठीक है? Next, when it is moderately active, at that time it is simple cuboidal, simple cuboidal epithelium, uh, moderate amount of colloid is present. And when it is highly active, okay, at that time simple columnar epithelium and very less amount of colloid is present. Now the question is, why the amount of colloid varies and what exactly is this colloid? Okay, now this colloid are nothing but thyroglobulin. The colloid, as I said, uh, pink color appearance, okay, homogeneous in nature. Okay, here I have mentioned. Now in this diagram you can see different types of follicles are shown. Okay, now in the follicle, first of all, see, this is highly active follicle, then this is moderately active follicle, and this is inactive follicle. If you take the example of inactive follicle, the lining epithelium is simple squamous epithelium. Huge amount of colloid is present. Next, if you see this one, this is moderately active follicle. इसमें lining epithelium कौन सा है? Cuboidal, simple cuboidal epithelium. Moderate amount of colloid is present. And if you see this one, highly active lining epithelium, simple columnar epithelium. Very less amount of colloid is present. ठीक है? So another question is, what exactly is this colloid? Okay, actually it is made up of thyroglobulin. It is a glycoprotein, and these are precursor of T3 and T4. Okay, when iodine is attached with this thyroglobulin, okay, after that further processing are done, and then these hormones are produced. Okay, so these colloid are nothing but the precursor of T3 and T4. Okay, next on the either side you can see the in case of thyroid gland, first, as I say, thyroid gland is covered by connective tissue capsule. So first there will be capsule, which is made up of irregular connective tissue. From capsule we are getting connective tissue septa. Okay, so connective tissue from capsule, the connective tissue septa will go into the substance of the gland and will divide it into numerous lobules. Okay, like this numerous lobules will be there. Okay, now if you take one particular lobule, if you take any of the lobules, then what you will see? First of all, through the connective tissue septa, the blood vessels from exterior, it is going into the gland. Next, if you are taking one particular lobule, then you will see that it is made up of follicles. Okay, it is made up of multiple follicles. Okay. Now, in between, in the follicles, what we can see? Inside the follicle क्या रहता है? Follicular cell. Inside each and every follicle, what we can see? Follicular cells are there. What else? Colloid plus colloid. Now what I have mentioned about the colloid, amount of colloid depends on the activity of the follicle. Okay, so amount of colloid okay, so follicular cells are there. So about the follicular cells already I have told you, different types of follicular cells are there, starting from simple squamous to simple columnar epithelium, and the amount of colloid and the type of follicular cells, it depends on the activity of the follicle. Okay, three types of follicles are there. What are they? Active, yes, moderately active, and inactive. Okay, three different types of follicles are there. So in case of active, which lining epithelium is there? Simple columnar. Here moderately simple cuboidal, and here simple squamous. Okay, so these are the three different types of lining epithelium. And what about the colloid? Here very less amount of colloid will be there. here moderate amount and here huge amount of colloid are present. Okay, so as I said, colloid में क्या रहता है? Thyroglobulin, okay, which is nothing but a glycoprotein, and these are the precursor of T3 and T4. Okay, from here only the T3 and T4 hormones are produced. Okay, now so all those things I have discussed. Next, suppose one follicle is there and another follicle is there. In between two follicles, the interfollicular space is present. Now, in the interfollicular space, what is there? What is situated in the interfollicular? Yes, blood vessels are there, nerves, lymphatics, all those things are present. Apart from this, specialized cells are present, and these are called as parafollicular cells. Okay, parafollicular cells. Okay, parafollicular cells. Now the question is, so follicles, different types of follicles, activity of the follicles I have mentioned. Now one thing you have to keep that in mind, the activity of the follicles, it depends on one particular hormone, and that is called as TSH, thyroid stimulating hormone, which is secreted from pituitary gland. The other name of pituitary gland is hypophysis cerebri. Okay, please keep that in mind. ये जो active, moderately active, highly active follicle है, वो कैसे होता है? Based on the hormone secreted by pituitary gland, that is TSH, thyroid stimulating hormone. ठीक है? So about the follicular cells already we have discussed. Now, if you see the EM structure of the follicular cell, okay, if you see the EM structure of the follicular cell, shows apical microvilli are present, abundant granular ER, prominent supranuclear Golgi complex, lysosome, microtubules, microfilaments, all those things are present in the EM structure of follicular cells. Okay, EM structure of the follicular cell. Now the question is, how you will differentiate between the follicular cells and parafollicular cell. Parafollicular cells, as I said, in between the follicle, the interfollicular space जो रहता है, in that parafollicular cells are present. ठीक है? Parafollicular cells, if you see, mostly they are in the form of cluster. Okay. Now, how you will differentiate between follicular cell and parafollicular cell? No, not exactly. Based on the staining difference. If you see the follicular cells, okay, they are basophilic in nature. So if it is basophilic, it will be darkly stained. And if you see the parafollicular cells, okay, they are pale staining. So like this, we can differentiate between the follicular and parafollicular cells. Okay. So here you can see parafollicular cells. First of all, it is polyhedral oval eccentric nucleus. Okay. Now these cells are embedded within the follicle or in between the follicles. Now this C cells or parafollicular cells shows numerous secretory granules. Okay, and as I said, for identification they are pale staining. Okay, and in case of follicular cells they are darkly stained. So like this we can easily differentiate. Next, what is the function? Now the follicular cells secrete hormone, that is T3, T4, already I have mentioned earlier. Okay, that is thyroxine, this T3, and what about the T4? Tetraiodothyronine, that is the T4, which is secreted by the follicular cells. Now what about the parafollicular cell? Parafollicular cell secrete the hormone, and the name of the hormone is calcitonin. Okay, it is calcitonin. Now, if you see the function, so T3, T4 जो होता है, T3 and T4, it controls the metabolic rate of the body in case of normal adult. But in case of childhood and during fetal life, this T3, T4 promotes the growth of the central nervous system. Okay, and calcitonin, what it will do? It will decreases or it will lowers the blood calcium level. How it will lowers the blood calcium level? By increasing the calcium deposition in the bone and inhibiting the bone reabsorption by osteoclast. These are the mechanism by which it will decrease the calcium level in the blood. Okay, so that is about the parafollicular cells. This already I have shown you. Yeah, this is the schematic diagram of the thyroid gland, ठीक है, which you have to draw in the practical notebook. तो इसमें क्या दिखाई दे रहा है? First of all, you can see what is this? This is the connective tissue septa. Okay, this is one septa. This is connective tissue septa. In between the septa, you can see this is one lobule. ठीक है, this is one lobule. Now inside the lobule, what is present? Follicles are there. So see, these are the follicles. ये देखो, these are the follicles present inside the lobule. In the connective tissue septa, you can see blood vessels are present. So these blood vessels, these are present in the connective tissue septa. Now, so this is one lobule, this is one lobule, this is one lobule. Okay, so in the lobule mainly you can see the follicles. Next, in between the follicles, in the interfollicular space, you can see the parafollicular cells also. ठीक है, ये देखो, these are the parafollicular cells. Now, in order to identify the parafollicular cells, what you have to do? You have to do, yes? No. In order to see the parafollicular cells, staining is there. Without staining, you can't see. You need high magnification. ठीक है? So see, this is the high magnification image of this particular area. So first of all, you can see the follicle. ठीक है? This is the follicle. This is one follicle. This is one follicle. ठीक है? Inside this, you can see colloid is present. Now these are the follicular cells. See, these are the follicular cells. ठीक है? तो ये एक follicle है, ये एक follicle. In between that, you can see the interfollicular space. इसमें blood vessels दिखाई दे रहा है. As you can see, the blood vessels are present. Along with that, you can also see the parafollicular cells. Now concentrate on the parafollicular cells and concentrate on the follicular cell. See the staining difference. Okay, see, these are pale staining, these are darkly stained. So like this, easily you can identify which are follicular cells and which are parafollicular cells. So identification of the thyroid gland, okay, is not that difficult, it's simple. So that is all about the thyroid gland. Next, here you can see low magnification image of thyroid gland. This is high magnification. Okay, so what you have to identify? First you have to identify a particular lobule. ठीक है, उसके लिए क्या करना है? First you have to see the septa, connective tissue septa. In between them, you can see the lobule. Lobule के अंदर क्या देखना है? Follicles. Follicle के अंदर क्या रहता है? Follicular cells and colloid. तो based on the colloid, you can judge which type of follicle is this. ठीक है. Next, in between the follicles, cluster of cells are there, which are the parafollicular cell. Staining difference also I have mentioned. Like this, you can identify. Okay. Next is the parathyroid gland. Okay, so there are two pairs of parathyroid gland which are situated on the posterior border of the two lateral lobes of thyroid gland. Okay. Here also, in each and every gland, connective tissue capsule is there. Okay, as you can see, each and every gland connective tissue capsule is there. From connective tissue capsule, septa is going, okay, and dividing it into lobules. But in this case, the lobules are not complete. It is incomplete in case of parathyroid gland. Okay. Next, within the gland, a network of reticular fibers are also there, which are supporting the cell, and many adipocytes or adipose tissues are also present inside the parathyroid gland. Okay. Next, the parenchyma of the gland are made up of two different types of cells, and those cells are in the form of cords, and in between them you can see the sinusoids. That is the important structure for parathyroid gland. So this is the posterior view of thyroid gland. ठीक है, तो thyroid gland का, see, this is one lateral lobe, this is one lateral lobe. तो lateral lobe का जो posterior border है, इसमें देखो parathyroid gland रहता है. Okay. So this is the area where parathyroid glands are situated. Now parathyroid gland, you can see surrounded by thin layer of connective tissue capsule, same like the thyroid one. Septa is extending, but it is dividing the gland into incomplete lobules, not complete lobule, poorly defined. Next, glandular parenchyma is richly supplied with numerous sinusoid. जो parenchyma होता है, in case of parathyroid gland, two types of cells are there. Here follicles are not present. Instead of that, two different types of cells are present, which are in the form of cords, and you can see the sinusoids. Wait. Now, as I said, two different types of cells are present. So what are the two different types of cells? Chief cells and oxyphil cells. These are the only two types of cells present in the parathyroid gland. Okay? Now the question is how to identify chief cells and how to identify the oxyphil cells. Chief cells are also called as the principal cells. Most number or numerous cells are present, means number of chief cells are more as compared to the oxyphil cells. If you see the shape, it is polygonal shaped, centrally located nucleus, dark staining cytoplasm. Please remember this particular point. They have dark staining cytoplasm. Okay. Now, depending on the quantity of glycogen accumulated and lipofuscin is a yellow pigment inside the principal cells or chief cells. It is of three types, as you can see: light, dark, and clear cells. Okay. Under electron microscopy, chief cell shows abundant rough ER, Golgi apparatus, and secretory vesicles. Now, the hormone which is secreted by chief cells or oxyphil cells, these are called as PTH or parathyroid hormone, or also called as parathormone. इसका function क्या है? It will increase the blood calcium level. Calcitonin will decrease, this will increase. Like this, the balance is maintained. Now the question is how it will increase the blood calcium level. Yes, there are some mechanism. As you can see, it tends to increase the serum calcium level. How it will increase? By increasing the bone reabsorption, okay, through stimulation of osteoclastic activity. Next, calcium reabsorption from the renal tubules and calcium reabsorption from the gut. Means whatever we are eating, from that the calcium reabsorption which is taking place. So all those things will be increased by the help of this particular hormone, parathyroid hormone or parathormone. Like this, the calcium level increases. Okay? So both the hormones which are secreted, that will balance the blood calcium level. Next, another type of cells are there, that is the oxyphil cells. Okay? So number of oxyphil cells are less as compared to the chief cells or the principal cells. They appear in the group or single. Under light microscopy, they are eosinophilic. Okay, eosinophilic means you can say pale staining. Next, these cells do not have any secretory vesicles. Okay, they don't have any secretion, and if they are having any function, that is still unknown. Okay. Another important point, if you see the oxyphil cells, the size of the oxyphil cells are larger as compared to the principal cells. Okay, so like this you can identify the principal cells and oxyphil cells. Now this is the diagram. This is the schematic diagram, and this is the original diagram, low magnification of parathyroid gland. This is the drawable diagram which you have to draw. So what you can see over here, first of all you can see this is the capsule, connective tissue capsule. In the connective tissue capsule you can see blood vessels are present. Okay. Next you can see septa is extending, but the lobules are poorly defined. That means incomplete septa cannot divide it into complete lobules. Next, inside the incomplete lobules, what you can see, only two types of cells are there. Here follicles are absent. What are the two types of cell? Yes, oxyphil cells and the principal cells. सबसे ज़्यादा कौन सा cells होता है? Principal cells. Okay. Yes. How to differentiate between these two cells? Staining property. तो ये देखो, ये जो cells है, these are the chief cells. तो chief cell जो है basophilic in nature होता है. You can say it is darkly stained. Next, these are the oxyphil cells. See, these are the oxyphil cells. Oxyphil cells का eosinophilic stain होता है. Cytoplasm eosinophilic होता है. That's why you can see pink color appearance हो रहा है. And it is larger and number is also less. Now, along with the chief cells, okay, along with the chief cells, you can see the blood vessels are also present. And here it is denoted as sinusoids. Okay, because यहाँ से secretion होता है, so that it can go to the bloodstream directly. Okay, and as I mentioned earlier, in case of parathyroid glands, you can see numerous adipocytes. Can you identify the adipocytes? These are the adipocytes. Here also, here also, here also. Okay, so these adipocytes are also there inside the parathyroid gland. Okay, so identification of parathyroid gland is also simple. It's not that difficult. Only two types of cells identify करना है, and before that you have to identify the capsule, incomplete septa, and then two types of cell. Okay, that's all about the parathyroid gland. Okay, So we'll go to the lab and then we'll see the slides.